And Logan has helped us review our epidemiology grants over the years. And with that, I want to thank you for joining us, and I'm going to turn the mic over to you. So thank you, Logan.
My pleasure, and thank you for having me. If I were to retitle this, I would call it "Why Do Kids Get Cancer?" Whoops, I gotta forward. This time it's epidemiological. That's because I volunteered for this series a couple of months ago when there was a talk, a very nice talk, entitled "Why Do Kids Get Cancer?" Which I found provocative, but it was very much the oncologist or the molecular biologist perspective. And what do I mean by that? So, to most oncologists, the cause of cancer is the mutation in DNA in a cell that inevitably turns the cell into misbehaving, that we call cancer. But that to an epidemiologist is not satisfactory. And at the time I raised the question, for instance, we know that 90% of Ewing sarcoma has the EWS Fly1 fusion oncoprotein. And many times you hear, well, we know what the cause of Ewing sarcoma is. It's EWS Fly1. And that's certainly true, that if you have EWS Fly1, the cell will turn cancerous. But in recent work by my recently graduated doctoral student, Brandon Diesner, we showed that the rate of Ewing sarcoma, unlike osteosarcoma, differs by race ethnicity, and this is controlling for socioeconomic status, so probably has something to do with differential genetics. The main point being that simply knowing what the somatic mutation is that leads to cancer is not enough to tell you what's cause. Rather, cause has to include cellular context and I would say germline genomics. There is a tissue context or microenvironment, and then we get to the organism, the person, and their inherent factors as well as their behaviors. There's their immediate environment, which in which they operate, in which contributes exposures, and then there's much larger global type exposures or area level exposures that everybody in a region might be exposed to. And I think tumor biology is mainly concerned with the sub organismal level of exposure or explanation, starting with DNA, the cell, and the microenvironments, and maybe the organ. But epidemiology, which is my field, is going to be concerned with everything from subcellular, molecular, all the way up to population level exposures. So having settled on a title some months ago, I found that I really backed myself into a corner as far as the content. Why do kids get cancer to an epidemiologist is an incredibly expansive topic. Honestly, the talk has evolved into more of epistemology than epidemiology, meaning how do we know what we know about the causes of pediatric cancer. So I have to give you a short lesson in epidemiology and causality. And then I will talk specifically about what is possible to understand in pediatric cancer because we are limited always by sample size and the rarity of pediatric cancer. And then finally, I'll deliver in a extremely glossed over version of what the title promises, which is the relative contribution of genes and environment in causing childhood cancer. So first of all, epidemiology has a dictionary which talks about the properties of a cause. Actually, causality, thoughts about causality go back many centuries, but the modern thought starts with David Hume. And when we talk about causality, we generally have to talk about in epidemiology the Bradford Hill criteria. Oh, one more slide. We also talk about causes being necessary and sufficient. So a necessary cause has to be present in order to cause disease. Sufficient means that the exposure alone can cause the disease without any other input. And those are precious few. I would certainly agree that ionizing irradiation it can be a sufficient cause by itself. Asbestos causes mesothelioma and may be the only cause. However, it's very rare to have causes which are both necessary and sufficient. And in fact, you can have causes that are neither. And most diseases, including pediatric cancer, are multi-causal. Can't delve too much into the philosophy behind this, but that will give you a 5-second version. I also want to distinguish between a risk factor and a cause. Honestly, epidemiologists as well as the general public will talk about this loosely, but there is a difference. A risk factor, let's take for instance birth weight, which is associated with most pediatric cancers. It predicts the occurrence of disease. So children with higher birth weight have a higher risk of cancer. But birth weight itself is a mixture of genetic potential for growth, maternal behaviors, for instance, smoking can depress birth weight. It also predicts future growth. And so it's not the cause in and of itself, but a marker for cause. So risk factors, when we speak loosely, can include both those that are causal and those that aren't. A cause, which we take great care not to call something a cause until we are quite sure, is something that meets the causal criteria. And then I will also add that we have different classes of epidemiologic exposures in childhood cancer. We tend to say exposures because epidemiology started out looking at things that were clearly people were exposed to, like cigarette smoke. But you can also speak about an endogenous or inherent characteristic like birth weight as an exposure, and DNA, genetic variation also as an exposure. So when we say exposure, we really just mean some factor with which we're trying to distinguish risk of disease. So the oncologists or the molecular biologists may study cells or non-human organisms, but epidemiologists will always study populations of people. And I will get to the study designs that are available to us, but always in the back of our minds are the Bradford Hill criteria for causation. Whether they're explicitly cited or not, virtually every paper that is epidemiologic in nature is going to reference these. And so I'll just go through briefly to orient those of you who are not epidemiologists. First, and these are not a checklist, but characteristics of causes. So number one is strength of association. The larger the association between a exposure and disease, the more likely it is causal. All right, so pack-a-day smokers have many multiples times the risk of lung cancer, and the reason is that this is a criterion is that it would be hard. You would be hard pressed to find a confounder or some other associated factor that could possibly be explaining that association, it would have to be very strong itself and probably would be obvious. Secondly, consistency. Individual studies have their own problems, but if we see an association repeatedly in different contexts, areas, different scientists, different study designs, then we start to believe it. Specificity is take it or leave it. It is true that asbestos it causes mainly mesothelioma, and that's extremely specific. But if we were to talk about smoking, of course, that causes many diseases in many organ systems. And so it's not a necessary. Temporality is the one criterion I will call metaphysically necessary. And that's because if your exposure does not precede the disease, clearly it cannot have caused it. But surprisingly, we still struggle with establishing whether an exposure was before a disease. Biologic gradient, also known as dose response, should be fairly obvious. If you give more of the exposure, you get more of the disease. That is the last of the major clues. And past that, we have plausibility, coherence, and analogy, which I think over the decades have shaken out as the least solid of the criteria, because they depend so much on which evidence you include in the broader universe of scientific evidence. Experiment, of course, is very important, but not always possible in people, in populations of people. And then lastly, some people will add reversibility. If you take away the exposure, does the disease reverse? But obviously, let's just take smoking and lung cancer. Once the lung cancer is progressed to clinical attention, taking away smoking doesn't stop the disease. So the driving force in what's possible to look at to do in pediatric cancer is the rarity. So pediatric cancer is 1% of the total cancer burden. And this often limits us to case control studies, which I'll explain in a minute, and at that, pretty small ones. I would add that that was 1% of total cancer, but then, of course, pediatric cancer is itself very diverse. And even within these categories, leukemia, ALL, for instance, has splintered into dozens of molecularly defined subtypes, which with quite a bit of evidence that there's etiologic heterogeneity, meaning that each of the subtypes may have its own unique set of risk factors, somewhat overlapping, but this is our challenge as pathology and molecular diagnosis improves over the years. Our rare diseases get splinter into ever more rare diseases. So the designs that are available to us, there are 4 basic epidemiologic study designs, starting with the ecologic study, which is not mean much to the layperson, but it means basically that we are looking at group rates. We are not looking at individual data, but trying to find a correlation between what's happening in a total population versus a total population exposure. So here's a study looking at gamma irradiation, which is an ambient form of irradiation. I believe this was in Germany, and showing that the rate of leukemia correlates with estimated gamma radiation. Ecologic studies are sometimes very informative for hypothesis generation. Here's an old set of data where I ordered the rate of leukemia, ALL, at the young ages. and highlighted those countries that reported different populations. And you'll see that in the US, white children have more than double the rate of ALL as do black children, yet live in the same country. I acknowledge that immediate home environment may well be different, but we see the same pattern in other countries. So in New Zealand, the not Maori and non-Maori have widely divergent rates. In Singapore, the Chinese and Malay have very divergent rates. And even in a country, Germany, back when it was East and West Germany, so the same ancestry, but divergent environments, we see a difference in rates. Recently, we looked at specific Asian ethnicity in the US cancer rates and compared that to the country of origin. And you can see on the left, we didn't see much correlation between rates for ALL, but quite a high correlation for rates of lymphoma, which was mainly driven by South Asians, Indian and Pakistani, who seem to have a high rate of Hodgkin's lymphoma, both in the country of origin and in the US, and incidentally in the UK. And so that's been attributed to high carriage rates of the EBV virus. So ecologic studies do give us fodder for further study, but aren't themselves definitive or ever giving us solid evidence of causality. One more example, this was buried in a paper of ours last year looking at 25 years of IARC data on cancer incidence. We classified countries not based on region, but based on whether they changed their Human Development Index countrywide over time. And interestingly, for ALL and to some extent for AML, those countries who increased in their Human Development Index, so in other words, were becoming wealthier, more educated, and materially better off, we did see increases in the rate of both ALL and AML. But there's a hard limit to what ecologic studies can tell us. And now I'm jumping to the best class of epidemiologic evidence, which would be randomized studies. So we flip a coin and assign exposure to people, and then follow them for the outcome. And this is considered the best class of epidemiologic evidence, because when you flip the coin, you make the two groups equal on average. So half the smokers will be assigned vitamins, the other half will not. Half the tall people, half the short people, etc. The only problem being, two problems for pediatric cancer. One, the rarity of the disease means we can't possibly enroll enough children and randomize to follow over time. And then secondly, some things are unethical to random. We would never decide to randomize pregnant women to smoke. And some things can't be randomized, such as, or not easily, such as body mass index. So there has not been, to my knowledge, any randomized study for prevention of pediatric cancer, except I'm sure someone will pipe in the context of predisposition. So the cohort study is the natural analog to randomized studies. So you start with disease-free people and determine their exposure in its natural state, and then follow people over time to figure out who gets disease. It has the advantage of clearly establishing temporality. And this is getting into some epidemiologic niceties, but if you confess to smoking in a cohort study, you won't be saying that with foreknowledge of disease. In other words, we have a lot fewer worries that the disease is causing people to report things differently. But again, the problem in pediatric cancer is that of rarity. There is an effort for existing pediatric cohort studies to pool them. It's called the International Childhood Cancer Cohort Consortium, or I4C. And they have assembled a fairly large number of children who've been followed either from pregnancy or from birth or at some point in childhood. And you can see they've amassed a large number of children. And, these are mainly questionnaire studies where the mothers have been questioned and followed over time. But you'll see that even with 183,000 children, they've ended up with about 700 cases total. Once we've split those cases into the many different types of pediatric cancer that we're interested in, the number for looking specifically at each cancer is fairly low. So, there is ultimately a hard limit to how much this kind of cohort pooling will tell us. Although they've certainly had very interesting results. Here's a study looking at the so-called photoperiod. So when a child is born, there's a process of programming them into the quote normal day for where they're born, and that's the photoperiod. And what you can see is that the third trimester photoperiod is slightly predictive of total cancer incidence. More often, we epidemiologists have been able to put together cohort studies using registry data. I have chosen to highlight the United States rather than Scandinavia, but Scandinavia is the epicenter for registry-based studies, so-called heaven for epidemiologists, because everybody has a tracking number, and the National Health Service records are made available to scientists. In the US, it's a lot more cumbersome process, but it can be done. So for instance, last year I published this. We linked a list of kids conceived in vitro to the birth and cancer registries of 14 states, and ended up with 275,000 kids conceived by IVF, and compared it to 2.5 million children in order to achieve sufficient numbers to compare cancer risk in kids conceived by IVF versus naturally. But you'll see even then, putting together several million children, we don't have overwhelming numbers of cases to point to. I would add that for those of you in the audience who may be concerned about hearing this, we mainly found that a very rare pediatric liver cancer, hepatoblastoma, was associated with IVF, while most other cancers showed no association. I'd also like to give a shout out to my dear colleague at Baylor College of Medicine, Philip Lupo, who put together a landmark study looking at 10 million US births, where he had linked the cancer and birth defects registries as well as the birth registries in several states, and showed definitively that particular classes of birth defects are related to particular cancers in much finer detail than anyone had done before. And again, this is what we could accomplish by linking together registries. But ultimately, there's only so much data you can wring out of a registry. We haven't, for instance, registered diet. We do not get good information on, say, smoking on birth certificates or alcohol use. And ultimately, the vast bulk of evidence that we have about what causes pediatric cancer are going to come from case control studies. Now, case control studies are can be done well, but also are easy to do poorly. And that's simply because of the way we have to go about it. So in a case control study, which is, canonically what you use for rare disease, and pediatric cancer certainly applies, you start with cases and you carefully get a selection of controls, and you ask about exposure in the past. And there's a number of things that can go wrong or cause case control studies to be invalid or have invalid results. Number one, who gets into your study? It is more difficult to get controls to participate than cases. The cases are motivated by disease to participate, and controls may have no particular motivation. In fact, we've documented falling participation over years. Secondly, even if you had perfect participation, it can't help but be the case that cases, or in our studies, the mothers or fathers who are answering the questionnaire know that the child got cancer, and therefore their answers may be colored by that knowledge. So for instance, if we ask about smoking during pregnancy, that's clearly going to lead the respondents to think, well, was it my smoking that caused my child's cancer? And we may not get the correct answer from inquiring that way. But the vast bulk of studies are conducted that way. I would add that the studies tend to be fairly small. So this is a meta-analysis from the CLIC consortium I lead and I'm going to describe in just a minute. But here we were looking at parental age as a risk factor for ALL. And you see New Zealand clocks in with a grand total of 90 cases, and Germany with 583, and in fact, an old study of in the US, which I steward but was conducted when I was in high school, is still the largest. We have to put these studies together in order to get precise results. So I did want to include another shout out to the CLIC consortium. We are the world's leading authority on the causes of childhood cancer and leukemia. And it wasn't quite ready to debut today, but next week we will go live at click.ngo for the public's website. The investigators' website is already live, hosted at IARC, and that's click.iarc.fr. And the reason that CLIC exists is to overcome the problem of small numbers. And so I'll just show you briefly a demonstration of the power of that. So cesarean section, of course, is very common, and there's increasing concern that it's not optimal for the subsequent health of children. In CLIC, we pulled together studies including several thousand cases of pediatric ALL and AML, as well as controls, and we found no overall association with cesarean delivery, but we did find a very slight but pretty certain association with pre-labor cesarean delivery. And if anyone's curious about our hypothesis behind that, I'll be happy to expand when we get to questions. But the smaller the effect, the harder it is to find. And so individual studies may never have found this 23% increase in risk, but CLIC, with its thousands of cases and controls, can find those kinds of risk factors. I would add the other theme throughout the last decade or two is taking the existing research and synthesizing it. So Click pools individual studies, but others can mine the existing literature. And here, colleagues in Italy looked at studies of air pollution, of road traffic density, and estimates of benzene exposure from air pollution in relation to ALL and AML. And they found some clear evidence of dose response, suggesting that at least a portion of pediatric leukemia is due to air pollution and specific components therein. The other class of case control studies which have been enormously productive are genome-wide association studies. And this basically means taking cases and controls, dropping their normal DNA onto a genotyping chip, which will look at hundreds of thousands to millions of genetic variants and compare them between groups to find variants that raise the risk of pediatric cancer. And although case control studies for environmental or exogenous or endogenous risk factors can be prone to bias, most people don't participate or in a study or donate their blood or spit because of one variant out of 3 billion in their genome. And so doing case control studies like this is relatively robust to the kind of validity problems that plague other case control studies. So that's in brief what the epidemiologic evidence looks like, or how we generate it. And now I'm going to just give, like I said, an incredible gloss on what we know about the causes. I will say that for fairly obvious reasons, we know a lot more about what causes or does not cause the more common pediatric cancers. So the leukemias, CNS, and lymphomas, and much less about what causes the uncommon cancers. So Ewing sarcoma or germ cell tumors. And I would add that in the common cancers for which we have a lot of evidence, when we see a null association in data synthesis, that is evidence of absence, meaning we have looked with enough times, with enough data to be able to say definitively there is no association between this putative risk factor and ALL. But for some of the less common cancers, we clearly have an absence of evidence for many cancers. And there, the only solution is to increase the amount of data to be able to say what the causes are or even what they aren't. But this is from a review of ours on the causes of ALL. Specifically, we were trying to make the case from the literature that the causes of ALL are heterogeneous depending on subtype. But what I really wanted to show you in this graph is that there are very few strong environmental risk factors for pediatric pediatric ALL. And I should say this is not just environmental, but also kind of endogenous or inherent risk factors. So just to orient you to what epidemiologists call strong, an odds ratio of 1 indicates there's no association between exposure and disease. An odds ratio of 2 means that children who are exposed have twice the risk of ALL versus those who are unexposed. And 2 is generally the consensus dividing point for what's considered strong epidemiologic association. And between 1 and 2, maybe you could add some adverbs for moderately strong. then verging into weak down here. And what you can see is clearly most risk factors are well under 2. And they're well under 2 with confidence intervals that are very tight, meaning that we now know that residential pesticide exposure raises the risk perhaps 30%, maybe 20 to 40%. And so it is likely a cause of ALL, but it's not a strong cause. And across the board, it is the case that there are no strong environmental risk factors for pediatric cancer, apart from a few niche exposures, for instance, prenatal medical irradiation, or postnatal for that matter. But thankfully, the recognition that prenatal or early childhood irradiation causes pediatric cancer came many decades ago, and the ethos is to reduce that exposure. Here was a review that Philip Lupo and I published this summer in Cancer Epidemiology, Biomarkers and Prevention. And I would say that this graph, which is supposed to read somewhat as a heat map, summarizes the level of evidence and the strength of association between a number of putative risk factors and some of the most common pediatric cancers. This was, I would say, semi-quantitative assessment. It was based on a sample size of 2 leading pediatric cancer epidemiologists taking their best stab at the level of evidence and the degree of association. But I think we'd find a consensus of scientists agree with our assessments. On the other hand, the genetic studies have been bearing fruit for the last decade. So here we have the polygenic risk for BALL, much of this generated by Jun Yang at St. Jude or Richard Holston in the UK. And although you don't see it in this graph, there are somewhere between 8 and 12 variants, common variants that increase or decrease the risk of BALL. And if you put them together, you weight them by the size of effect, you can create a polygenic risk score. And here you see the referent is the median of the score. And for those in the top 1%, they have 5 times the risk of ALL versus the median. If you actually compared it versus people at the bottom of the scale, it's more like 9 or 10 times. For those who have the lowest, are on the left-hand side of the polygenic risk distribution, and those on the upper side. And I want to put this in perspective. For prostate cancer, which of course is magnitudes more common, they have a polygenic risk score of dozens or hundreds of variants, which describe maybe a 5-fold risk in prostate cancer. And so the rule in pediatric cancer has been that common variants have a larger effect on pediatric cancer. I think that this went against most people's expectation, but nevertheless, it's the case, and it's why GWAS studies work in pediatric cancer. We could put together similar curves for neuroblastoma and Ewing sarcoma, which are the other 2 pediatric cancers with fairly mature common variant genetic architectures. And I'm sure as the studies come out in other types of pediatric cancer, we'll see something similar. Also, in the sequencing era arrived about 5 years ago with this paper from the Pediatric Cancer Genome Project, and just showing that for most pediatric cancers, the bad news genes, that is point mutations or other types of mutation in classic cancer susceptibility genes, only explain between 5 or 10% of most pediatric cancers. Although there are some cancers like adrenocortical carcinoma, for which the bad news genes explain a very large proportion of cancers. These are just looking at the known cancer genes, but there are, from sequencing, some surprises out there. So earlier this year, there was a fantastic paper showing that in one subtype of medulloblastoma, that which is has sonic hedgehog alpha mutations, 31% of those cancers have a mutation in a gene called elongator, or ELP1, which now describes a new cancer predisposition syndrome. That said, even describing the cancer a strongly determinative predisposition isn't enough to completely describe cause. So for instance, this is another recent paper from a group looking at the penetrance of Li-Fraumeni. Penetrance means that the percent of people who carry a genetic variant who will develop the disease. And even in those who have Li-Fraumeni syndrome, meaning that they have a vastly higher risk of both pediatric and adult cancer than the general public, only 40% of people with Li-Fraumeni go on to develop breast cancer, sarcoma, or other cancer. And so there must be other factors that go into causation of cancer in these individuals. Lastly, I again want to highlight some work that was published earlier this week from my now postdoc, Brandon Diesner, which was looking at de novo mutations as a cause of pediatric osteosarcoma. And we found the only gene that was repeatedly hit by de novo mutations was TP53, which underlies most of Li-Fraumeni syndrome. And the main finding was that, so it's no surprise that TP53 was related to osteosarcoma. But the main thing I want to point out is that a share of pathogenic mutations in pediatric cancer are de novo. In this particular case, almost half of the pathogenic mutations were de novo. And that means that we now can also study the causes of de novo mutation as an upstream risk factor for developing genetically associated pediatric cancer. Now we know what the main risk factor is for having a de novo mutation, and it's older paternal age. But even within that realm, there's more room to find out why do some children, are they born with a mutation that their parents didn't have? So much more to come. So conclusions, I think for those who haven't thought about causality before, just know that it has a definition and that there are formal criteria for assessment of causality. In pediatric cancer, the rarity very much drives what is possible for us to do. But we have nearly 40 years of epidemiologic study of causes. And so through data synthesis and especially pooling through the CLIC and I4C consortia, we are identifying both exogenous and endogenous and genetic causes. The genetic causes especially are becoming clear through GWAS studies and next-gen sequencing. And my overall assessment, if you pin me down, is that strong environmental causes are few. In fact, strong endogenous non-genetic causes are also fairly few, while genetic variants, both common and rare, are much more prominent. In other words, explain a larger proportion of cases. So at this point, I will pause for questions, but you're also welcome to email me with any. So I don't know, do you read me the questions, Jay, or do I read them from the chat?
Yep, I'll read them for you. It'll make it easier because they're spread throughout. Thank you very much. I found that really informative, and I learned a lot during your talk, so I really appreciate that. We're going to jump right into the questions. First question is, in your recent paper about childhood cancer progress and priorities with Dr. Philip Lupo, you mentioned that Mendelian randomization can use genetic variation to examine the effect of an exposure or another trait on disease in observational studies. Can you briefly provide an example and explain how this works?
Yeah, so we can't randomize genes, right? But every allele that you get, an allele is an alternate version of a DNA letter, okay, is essentially a coin flip. You know, if your father has AB genotype, you have a 50% chance of getting A, a 50% chance of getting B. If we're confident that we know what the genomic architecture of, say, BMI is, we can look at each of the SNPs that causes BMI and apply that to pediatric cancer. And, can't say in 30 seconds how the study design works, but essentially you are randomized at birth to a certain genetic potential for BMI. And you can apply that information to the GWAS data for pediatric cancer and come up with a causal statement about maternal, for instance, maternal BMI or the child's BMI potential and the cancer. That was the worst 30-second explanation of Mendelian randomization ever, but I'm happy to expand by email.
Okay. The next question is, how do we explain the discrepancy of occurrence in pediatric cancers between the industrialized world and the developing world?
That's a great question. And actually, I kind of recall the earlier slide I showed about air pollution. You know, we know that air pollution, it kind of has a U shape as societies get richer. So there's unindustrialized societies where there's not a lot of air pollution because there's not a lot of sources, and then there's middle income countries where there's a lot of sources as people get cars and factories are built. And then there's the end of the curve, which is the most developed nations where people have, excuse me, the economic luxury to start caring more about the environment and mitigating it. And so, you know, one reason that cancer may rise as countries industrialize is these exposures. Certainly, the population genetics don't change over time. So when we see a rise in countries, it's more attributable to environment. That said, sometimes there can be artifactual rises. So as countries get richer, they can also afford, say, MRI machines, and they will detect more indolent brain cancers than they otherwise would have. So like most questions, it has a complex answer.
A lot of moving parts and things to think about there.
For sure.
The next question says, could you touch base on the epidemiologic risk factors for CNS diseases such as medulloblastoma? And I think you did talk about that a little bit.
Well, so part of the problem, and maybe this theme didn't come through in what I was talking about, is a lot of the epidemiology for many cancers needs to be redone. So medulloblastoma is one of those cancers that has splintered into first 5 and now 7 or 8 subtypes. And in almost any pediatric cancer that we look at, when you start to look at risk factors in relation to tumor subtypes, you see a lot of what we call etiologic heterogeneity. And so almost all the epidemiology for medulloblastoma has been with the lumped variety. We need a whole new generation of studies that look at specifically within the subtypes. So I hesitate to make any statement about Medulo as a whole, because that research needs to be redone.
Wow. Wow, that's a lot of work to redo then.
Yeah, although I got 30 years left in my career, so.
But then you're talking about smaller groups also.
We are, and it also again, that points to the need for international collaboration, because even in the U.S., which is a fairly large country, there's only I think 400 Medulo cases a year. We really need to band together internationally to get enough sample size when we splinter these cancers.
So next question is, do you have hope that the NCI's Childhood Cancer Data Initiative will help you collect and analyze expanded, deeper data related to the causes of childhood cancer? And then follow up to that, are there things that advocates can do to help your efforts, especially related to data collection and aggregation?
Yes, and I'm on at least one of the advisory panels for the CCDI. I think the one thing CCDI is not doing is surveying parents directly, but they are making an effort to do the kind of data linkage studies that I advocated before. So they're going to pilot linking to birth registries, and although that amount of and possibly birth defects registries, the amount of data that's in there is limited, but there's still an incredible amount of information that we can squeeze out of that. They also are attempting to automate residential histories, and some things like, say, air pollution or proximity to agriculture, from which we can estimate pesticide exposure. We can gain a lot of that information from residents. And so the two things that they are doing that most help identifying environmental causes is linking to the data linkage to birth registries as well as residential history. And then I would add, anytime we drop a DNA on a GWAS chip or sequence it, that's an untold amount of information to mine as well. And the combination of the data linkage and the genotypes and sequencing will be very powerful. As for what advocates can do, to tell the truth, I don't know that there's a direct route from advocates to CCDI. But if someone from CAC2 or ALSF or some other advocacy organization would like to make their voice known, I think we could certainly get you in the room.
Great. We have got a ton of questions that have recently come in. There's no way that we can get to all of them here, but hopefully you could take a couple of more, and then maybe you'd be able to answer some for us offline. We could connect you with the people who asked them, or maybe we could even put together a blog post where we answer some of these questions. Absolutely.
I'm very happy to answer it. And I will also add that, when we debut the CLIC website next week, that some of our collective research will be, easily available to the public. But
okay. The next question comes from Barbara Rivera. She said that was really great, and she's curious about your hypothesis regarding pre-labor cesarean, I think it says, in cancer risk.
Yep. So this paper was written by one of my faculty members, Dr. Aaron Marcotte, and that originally when we wanted to look at C-section, we thought that it would have something to do with the microbiome. So when a baby's born vaginally, they gain the bacteria. And when they're born by C-section, they come out actually relatively uncovered with bacteria. And surprisingly, though, we didn't see any difference with any C-section. It was only when we looked at pre-labor that there was this difference. What is the difference between pre-labor and post-labor C-section? Labor involves stress. to both the mother and some to the child. In fact, there are studies showing that in the cord blood of children born with pre-labor C-section, you see much less cortisol than in kids born after labor. And actually, it goes with length of labor. Cortisol is the natural version of glucocorticoids, which are the backbone of ALL therapy. And so the hypothesis is that children that labor, it involves cortisol exposure, which is analogous to glucocorticoid exposure, and there may be a culling effect of pre-leukemic clones. So we do know that ALL and AML can start in utero, and there are circulating cells that are kind of halfway to leukemia, and it may be that lack of labor means that these cells do not get culled out of the blood population.
Do you have time for a couple more?
I do.
Okay. Next question from Philip Lupo. Little known fact. Hi, Phil. He owes me beer. He owes me beer.
He owes me beer.
Really?
Oh yeah, always.
Amazing. Were you surprised about your findings for de novo variants in osteosarcoma? And then did you think there would be novel de novo variants underlying risk? And then are there some next steps for this analysis?
Great question. I was surprised. We had seen in some of our data an association between osteosarcoma and rising parental age. And I already mentioned that older paternal but also maternal age can lead to more mutations in a child. Also, the worse a gene is or a variant is, the more likely that it appears only from de novo mutations. In other words, if a mutation is so bad that it means that a child can't grow up and reproduce, the de novo mutations will be the main reason that you see it in the population, right? Because otherwise they couldn't pass it on. So I did think we were going to see more genes that were repeatedly hit. In fact, that study design is especially good for finding genes with a small number of cases. That said, this study design was pioneered in autism, and in autism, the paradigm is that pathways are repeatedly hit, but individual genes may not be. It takes a lot of genes to create a brain because it's a very complex organ. But once you start to look at all the genes that create a brain, you can see that they are repeatedly hit by de novo mutations a lot. So I think we need to take this study design and expand it to more osteosarcomas and more of, frankly, all pediatric cancers. Because with sample size, we can start to slot these de novo mutations into pathways rather than individual genes.
Thank you. We're going to do one more, and then we'll get a list of the rest of the questions and put those together for you. One comment did come in from a parent who has an ALL. Child is 21 months old in Minneapolis, and they said they're happy to participate if you need them to answer any questions in order to help your studies. So we can connect you if that would be helpful. And then the last question we're going to do is from someone named Kelly. They said, great talk. They really enjoyed the overview of the aspects to consider. And they asked, what is your opinion on optimal sample sizes to perform case control and cohort meta-analysis studies, given the obvious limitations in availability of rare patients to study?
Well, meta-analyses mine the literature, so that's not a matter of sample size. When you say case control studies, if we were to start a new case control study today, it depends on the frequency of the exposure you want to look at. So I would not start a case control study to look at IVF, because that's about 1% or 1.5% of the of births, and so you would need a couple thousand cases to be able to look at that reliably. If you're looking at something that is more common, let's say maternal prenatal vitamin use, then you can get away with a smaller sample size. I think too that the sample size calculations and optimal sample size is a luxury of more common diseases or common cancers. For pediatric cancer, we don't have that luxury. Frankly, I think we should be enrolling every child in a study of the causes. And not coincidentally, the Children's Oncology Group has something called Project Every Child, which is not quite there for enrolling every child, but that is the aspiration. At least in the U.S., I think it's very, very achievable. Other countries we would like to join along, but that would be the ambition, is to have these epidemiologic studies on a continuing basis. But it's a tough case to make to grantors.
Yeah. I want to sneak one more question in here.
Yeah.
This is from Amy. Hi, Amy. Have any studies been done on twins, in particular, where you have one who was affected and the other healthy?
Yes, there have been. In twin research, and Amy, maybe you have twins, and that's why it's on your mind.
She does.
Twin research tells us about heritability. If you see identical twins are more concordant than fraternal twins, you can calculate a heritability from that. We can't really do that in pediatric cancer because let's say that, common SNPs double the risk of ALL. That means it goes from 60 in a million to 120 in a million risk. And so you would need thousands of twin pairs to be able to see the concordance. Now, so we haven't really seen a lot of heritability studies in twins for pediatric cancer because the incidence is just far too rare. On the other hand, for leukemia in particular, because it is a circulating cancer, we do see a higher risk of twin concordance. And it's not because of heritability, it's because monozygotic twins, 60% of them share placental vasculature. In other words, blood can cross from one twin to the other. And so Sir Mel Greaves in London was knighted partly on this research for showing that in twins that are concordant for ALL and some with AML, they have the same mutation, not just the mutation in the same gene, but the same exact breakpoint. In other words, the cancer, the leukemia started in one twin and it migrated to the other. So yes, there's quite a rich amount of twin research in pediatric cancer, but it is more about natural history than heritability.
And we saw him speak years ago at the Children's Hospital of Philadelphia. It was fascinating. And he was talking about where a lot of times they thought there needed to be one more thing happen that sort of triggered the leukemia to turn on.
Yeah, so the major lesion, let's say ETV6 RUNX1, has been shown repeatedly to be in utero, but then the secondary copy number alterations like Icaros deletion, etc., those probably happen postnatally, and that's why epidemiologists concentrate, childhood cancer epidemiologists concentrate on the period just before conception, pregnancy, and early childhood to try and figure out why kids get cancer.
All right, well, thank you so much. Thank you to everybody for joining us. I'm sorry that we ran late. Jen is going to put together the rest of these questions. Oh, and then Liz just sent in, do you need to monitor an identical twin if the other develops leukemia? She just...
That's a great question. And I think that since they have minimal residual disease, ascertainment for the twin with frank leukemia, they certainly at this point could detect the clone in the other twin, but I'm not sure anybody's pursued that clinically. But it's well within the realm of possibility.
Thank you. Thank you very much, Logan. Thank you for everybody for joining.